So we'll start the first session next on chemistry, and Tim is going to be chairing that session. So I'll leave it to you, Tim.
Great. Well, thank you for the transition, Serene. I'll say that I'm excited to be opening this first session about Dell chemistry and how libraries are built and made. I'll say I think that it's an attractive idea to be able to run these and produce these very large combinatorial chemistry libraries, and that can get you into a lot of trouble when it comes to applying Dell to drug discovery, and in the sense that you can make molecules through the combinatorial synthesis that have fairly poor physicochemical properties and aren't useful in serving purposes in the pharmaceutical sector. And so I'm super excited to hear Chris's talk here from X-Chem about how they think about designing Dell libraries and picking monomers to be included within the combinatorial chemistry synthesis procedures to assemble Dells that have good chemical properties and make good starting points for projects. So a little bit about Chris. He joined X-Chem in 2010 and is an associate director of discovery chemistry. He's had over 10 years of more than 10 years of experience at X-Chem, designing and producing DNA-encoded libraries, as well as supporting other aspects of discovery with Dell technology. With that, I'll let you take it over, Chris, and the floor is yours.
Thanks, Tim, for the introduction, and thanks for the other organizers of the workshop for including me. I'm going to share my screen here. It's not easy to follow Professor Neary, so I'm hoping to do a decent job. This into presentation mode. Okay. Can you see the slides? Okay.
Yes.
Excellent. So today I'm going to talk about some design concepts that we at X-Chem feel are important to make some high quality libraries. And this first slide right here is an overview of the platform. It's the process by which is familiar to other practitioners of the field, where we have at the far left-hand side of the slide, the design and synthesis of our DNA-encoded libraries, which are then used for the selection process, which is the next circle over. Of course, we need to take that output, run the sequencing, do the data analysis, and synthesize some representative compounds off DNA, and then test them in the assay of choice, and hopefully identify leads. That's the overall goal of all of our projects, is to identify some active and functional compounds to the projects. The talk today will really focus on the bookends of this process. So I'll be talking about the design and synthesis of our libraries, as well as the output from some of our most recent libraries. So here's I guess a rough representation or a crude representation of what a library member would look like in a DNA-encoded library, where the left-hand side is comprised of building blocks that are covalently linked. This would occupy sort of the warhead side of a library member. It's then linked to our headpiece, which is essentially the starting material on the DNA side of things. Then we have our DNA tags, which encode for the synthetic history of a library member. And in this representation here, we have the green, the pink, and the blue building blocks, all encoded by the green, the pink, and the blue pieces of DNA. Some of the key advantages over other encoding methods include the large split sizes that are supported with DNA-encoded libraries. We can incorporate over 10,000 building blocks in libraries. And another key point is, once we make a library, that library lasts for a very, very long time. So we can use a library in over 100,000 individual selection experiments. Two of the key components to sort of the warhead side of a DNA-encoded library are building blocks and chemical reactions. We need the building blocks, obviously, to make the warheads. And on the left-hand side of this slide, I have some colorful shapes to show you the types of building blocks that we use and incorporate into our libraries. You know, the bigger the circle, the more building blocks we have of those. So, you know, amines, carboxylates, aldehydes, you can get well into the 10,000s of building blocks available for those. There are some other building block classes that are less commercially available, but we try our best to incorporate as many as we can. Getting building blocks into our library or into our collection is an ongoing process. So every year we're just adding more and more building blocks to our collection. In addition to building blocks, we also invest in chemical reactions. So there's a number of chemical reactions that have been reported to be compatible on DNA, such as the ones listed on the right-hand side here. And we utilize many of these in our libraries, along with some of our proprietary chemistry that we develop in-house. About just over 25% of our libraries feature some proprietary chemistry. Now I'll go into some more detail on that a little later in the presentation. But I really want to focus on is sort of the left-hand side. Building blocks are the key to making quality DNA-encoded libraries, and I'll tell you why. This is an overview of our library deck. If you look on the left-hand side, there's the few charts there. And it just gives a couple examples of libraries that we have in our collection. We make libraries that have 2 cycles of chemistry, 3 cycles of chemistry, and 4 cycles of chemistry as well. And you can see as you get into libraries that have 4 cycles of chemistry, such as example 4 in the sort of the blue circle here, you can get some quite impressive numbers as far as your library size. You're talking about into the billions of compounds, theoretical compounds in your library. But along with that high library size, you get higher molecular weights. So if you look at example 4 here, yes, we can make a library that has 1.5 billion members in there. But if you look at the average molecular weight on the bottom here, it's around 750. So the compounds that are coming out of here are well beyond the Lipinski compliant parameters. And we acknowledge that there is cases where going beyond rule of 5 is warranted. But what we tend to focus on are libraries that use 2 and 3 cycles of chemistry to build our libraries. An example like that is example 1 in this graph. We've kind of blown out on the right-hand side an oxidiazole library that uses 2 cycles of chemistry, 2 types of building block classes. And in the box and whisker plots, you can see the property ranges for this library. So the average is in the middle of the block of the box. So for molecular weight, we're looking at an average molecular weight of around 385. You can see in the shaded boxes, sort of the majority of the compounds fall in some very attractive property profile. If we focus on 2 cycle libraries, how do we get the most out of that? How do we maximize the potential of a 2 cycle library? And it all comes down to the split size. It's the number of building blocks you can incorporate into your library for each particular cycle. We can operate at split sizes over 10,000, and this isn't a problem. The encoding capacity is well beyond the number of building blocks commercially available. So we don't have to make choices on building blocks based on the encoding capacity. For example, if you were to use a 10-mer piece of DNA to encode building blocks, you can encode 1.05 million building blocks just with a 10-mer piece of DNA. So we're not limited by the encoding capacity by any means. So what we do is we set up property and structural filters when we look at building blocks, and we buy as many as we can. We buy as many as the budget can allow. Obviously, we don't want to include panes or any other type of functional groups that one wouldn't want in sort of a hit or a compound to progress into a medchem campaign. And you can set limits on molecular weight, C log P, rotatable bonds, etc. And everyone has their own opinion on sort of what those limits may be. But there's a cost of focusing on building blocks and incorporating as many as you can, increasing that split size into that, the thousands or 10,000s mark. And that's property inflation. So this slide is showing primary means that are available from E molecules and enamine that are under 300 Daltons for molecular weight. On the x-axis is the number of building blocks. So this would be your split size. So we go from 0 all the way to 40,000 building blocks. And on the y-axis, we have average molecular weight. And what you can see is, as you get to split sizes or number of building blocks close to about 1,000, see a rapid increase up to molecular weight around about 125. And then as you get higher and higher split sizes into the 10,000s and 15,000s, you can see your average molecular weight does increase. But what you also notice on this graph is, yes, we may increase in molecular weight as we get more and more building blocks, but the cost to incorporating more and more building blocks becomes less as you go to higher split size. So as you go from 0 to 1,000 building blocks, you have a high sort of change in molecular weight. But as you go from 5,000 to 10,000 building blocks, your change in molecular weight is not that significant. So there's no extra penalty for going to higher split sizes. We can apply this rationale to C log P as well. This slide shows the same set of primary amines. The top graph is ordered by molecular weight. Ordered by molecular weight. The bottom graph is ordered by C log P. And what you can see is the same trend follows as what we saw before, is that as you get higher and higher number of building blocks, meaning your split size gets higher, we're not paying as severe of a penalty for incorporating more and more building blocks. But we do acknowledge, for C log P, the C log P of a building block may not correlate to the C log P of a final product, but we do see some correlation in our experience. It really depends on the types of chemistry you're using with those building blocks. So the conclusions we can draw from the analysis of the molecular weight and the analysis of C log P, we feel can be extended to other properties as well. So what does this mean for DEL library design and DEL synthesis? This means that we can get the most out of a DEL when we are BB inclusive. We want to make sure we can maximize the split size in our libraries. And there's a few things here in the sentence that I want to point out. By being BB inclusive, yes, we want to buy as many building blocks as we can that fit certain criteria, but they also have to work in the library, right? They have to be able to do the chemistry that we want them to do in order to make it into library. So all of our BBs go through a rigorous development and validation process to make sure they can undergo the chemistry we want them to do when we put them into a library. The figure on the right-hand side of the slide just shows sort of an example of sort of a BB validation. We'll take a model system, a system that is sort of mediocre as far as conversions, and test, in this case, all of our amines against that model system. And we can identify the building blocks that can perform that chemistry and those building blocks that can't, and sort of set thresholds and include building blocks that have high conversions into our library. By including building blocks that have high conversions, along with library purification, this can ensure that we have high fidelity DNA-encoded libraries. What do I mean by getting the most out of a DEL? There's a couple of things. One is diversity. So we feel that building blocks supply all the diversity we need for a library. This slide shows some random carboxylic acids from our collection. And you can see if you peruse the structures that there's a variety of shape, 3D content, electronic character, charge, etc. from just a sample of 30 or so building blocks. Now imagine making a library with 5,000 acids like this, or 10,000 acids like this. This is going to give us the diversity we need to cover as much space as we can and be successful in our projects. We're not the only ones that feel this way. Our thoughts are echoed in a recent publication about last year from some scientists at AstraZeneca. What they did was they looked at 10,000 compounds, small molecule compounds, that were made with easy chemistry, meaning they were just made from an amide bond formation. And they looked at 10,000 compounds that were made with 150 other chemical reactions other than amide bonds. And they looked at complexity, diversity, and novelty. And basically what they concluded was, it's not the chemistry of connecting these building blocks that's driving complexity and diversity. It's the building blocks themselves. On the right-hand side of the slide, I show a few graphs from their article, the top showing the complexity. If you look at sort of the complexity diagram there of the compounds made from simple amide bond formation versus the other 150 reactions, as well as the chemical database. you can see it's very comparable as far as the complexity of complexity of compounds you can make with those building blocks. Similarly with diversity, which is on the bottom part of this slide, if you look at the sort of diversity of the compounds you can make just with amide bond formation and versus the other 150 reactions, you see comparable results. The authors do not feel that we're losing anything by doing simple chemistry, but using a lot of building blocks to get that diversity. Another thing we gain to get the most out of a DEL by being BB inclusive is library fidelity. I kind of talked about this a few slides ago, but if you apply stringent validation criteria and you use chemistry schemes that are shorter, low cycles, two cycles of chemistry, high split sizes, you can get high fidelity libraries. This can pay dividends down the road. For example, 94% of our licensed compounds to our partners are the exact structure indicated by the encoding. So there isn't a need for elaborate follow-up campaigns to identify the actual actives that come out of a screen. Our DNA code is very predictive of the structure. Additionally, if you can synthesize a high fidelity library, high fidelity DEL, it can help drive machine learning endeavors. So we published this last year in the JMedChem with some collaborators with Google. We used our library screening data to support machine learning prediction models. So the high fidelity libraries that we can make is clean enough to do this with machine learning. So what we did was we used our selection output as the positive training examples. The noise would be the negative training examples. We built models, two different models, GCNN and the random forest model. We built some models. We can then predict, we can make predictions on virtual libraries such as MQL or large commercial databases. So predict which of these compounds might be active or might be a binder to the protein of interest. Acquire these compounds and test them. We did this for 3 different targets, which is shown on the bottom left-hand side of the slide: SEH, soluble epoxide hydrolase, ER-alpha, and c-KIT. And what these colorful bars show for each target is sort of the hit rate of identifying actives from the machine learning endeavor. So the lighter the blue shaded color, the weaker the active is. So you can see like white would be sort of not active. A dark blue in the case of SEH is 10 nanomolar. So you can see we had a high hit rate for a number of these targets. Not only did we find a number of hits, we actually identified a number of potent hits. And that's indicated in sort of the darker shaded regions of these graphs. Identified a number of compounds below 1 micromolar. The 1 micromolar mark is indicated by the dark black line here on each of these graphs. And these compounds just weren't similar compounds to the training examples. On the slide on the right hand, or the graph on the right hand side, this bar chart just shows sort of the similarity of the actives against the training set. And it's showing that not everything that we found that were active was just similar to the training set. We found things that were actually unique from these external collections that were found to be active. Another thing that high fidelity DEL can allow you to do is derive selection or information from the selection to help with hit optimization. So if you include as many building blocks as you can in your library, and you build a high fidelity DEL, you can get some SAR, if you will, out of the library. What that means is, you'll be able to observe trends in the enrichment data for structurally similar building blocks from the initial selection experiments. What I've shown in this slide here, just a handful of building blocks you can incorporate into your library, and you'll see the trends in enrichment to give you a sense of what the SAR might be demonstrating for binding for this particular enriched instance. And this type of information can help jumpstart your hit optimization program. It gives you some information right off the bat to help you optimize those hits. if we're BB inclusive and we want to synthesize high split, low cycle libraries, then we have to sort of change our mentality on how we design the schemes for those libraries. We have to design schemes that can deliver attractive property profiles when we utilize these large and complex building blocks. So if we have a lot of molecular weight tied up in the building blocks, we need to minimize the number of non-BB atoms. So what that means is you need to design libraries that don't have elaborate cores, and then you're being decorated with many cycles of building blocks. We don't need to have preformed cores. What we like to do, and what we would urge the industry, is to form scaffolds, i.e. rings, from the BBs themselves. Use the building blocks to help construct your I guess, core scaffold of the library. We have some examples of what I mean by that. So on this slide, it shows some libraries that we've made at X-Chem, some atom-efficient cyclization strategies. The top case, starting from an aldehyde on DNA, incorporate hydrazines to make pyrazoles. Going down the list there, we can make some aliphatic azocycles by using primary means as sort of a key component. And other libraries, such as oxidiazoles or other azoles, you can make from building block classes like carboxylic acids and aldehydes. And so we're incorporating atoms from these building blocks into the ring system that we want to make. We're not using capping chemistries. So these libraries tend to have very attractive properties compared to libraries where you're successively concatenating building blocks and then capping amines in various positions. Here's an example of a oxidiazole library, a 1,2,4 oxidiazole library. Has roughly about 2 million members. On the left-hand side is a spider plot, a radar plot, which shows the sort of the property ranges and averages for this library. And on the right-hand side is the box and whisker plots, which give you a better breakdown of where the bulk of the compounds lie as far as property ranges. What you can see, if I draw your attention to the sort of the box and whisker plots, is that the averages for a variety of properties are in a very attractive range. And even the majority of the libraries, which is sort of shown in the boxed colored boxes for each of these properties, are in ranges that would be good starting points for any medchem campaign. This library, or the data that you see, is derived from a random set of 6,000 fully enumerated library compounds from this library. In a similar vein, we can look at aliphatic azocycle libraries. Here's an example of one of those, where we incorporate 2 cycles of building blocks, high number of building blocks in each. We're getting about a 5 million member library. Again, showing the property ranges on the left and right-hand side. Here, again, the property ranges are in very good space for a medchem campaign to start. One key difference here is, before we were making an aromatic oxidiazole ring, here we're making sort of a more flexible sp3 rich azocycle. And so you can see that the fraction of sp3 character of this library has greatly increased. We kind of flip back to the previous one. Roughly the average FSP3 is around 0.3 for the oxidiazole, and here we're around 0.52 for this azocycle library. So again, high split sizes, low cycle numbers, using building blocks to create ring systems, cores, will give you property profiles that are very attractive. Here's a slide that shows some of the library outputs for libraries made, some of the recent libraries that we've made over the last few years. And I guess what I'll draw attention to in this slide is not only the potency of some of the hits coming right out of the library, single-digit nanomolar hits, double-digit nanomolar, but what we like to focus on is really the ligand efficiency of these hits. So we're operating in many of these projects where we're identifying hits that have ligand efficiencies of 0.3 or greater. Again, these are the starting points for a medchem campaign. And so obviously we want to be successful when we partner with our collaborators. We want to get them the best quality that we can right out of the bat. And giving them something like in this list here is a great starting point. On the right-hand side, just show some more specific examples of some recent hits coming out of our libraries. This top compound is from C-Kit, right out of the library. It's 3 nanomolar in a biochemical assay, gives you a ligand efficiency of about 0.43. We have a recent hit for ER alpha, again, single-digit nanomolar coming out of right out of the screen, ligand efficiency of about 0.39. And another example from a hydrolytic enzyme, again, single-digit nanomolar potency and a ligand efficiency, close to 0.4. Again, all of these data that you see in this slide are initial hits straight out of the library, what we would call first pass synthesis, initial hits that we would be given off to our partners for a medchem campaign. Oh, and the last thing I wanted to talk about here is the variety of targets classes. So you can see it's not just one particular target class that we're able to hit with these types of libraries. We have a range of target types, PPIs, cytokine PPI targets, kinases, ligases, GPCRs, nuclear receptors. So a variety of targets are able to be targeted with these libraries that give very ligand efficient hits. I'm going to spend the last few minutes talking about some examples, examples a little more in depth of quality hits coming out of our library. First example is a kinase project that I worked on a few years ago. This kinase was an essential component of the immune signal transduction pathway. A partner came to us wanting a selective inhibitor. There wasn't any known selective inhibitors known to date for this target, known in the literature. We ran a selection campaign with them, and we identified 8 selective submicromolar chemical series through our platform. And many of the series had ligand efficiencies greater than 0.3. I have one example here at the bottom left-hand side, comes from one of our azole libraries. It's a 2-cycle library. This compound was about single-digit nanomolar active in the on-target assay, about 8 nanomolar, shown on the curve on the left-hand side here. Did not hit the off-target, so this definitely matched the profile that the partner was wanting. Has a ligand efficiency of 0.39. The partner took this compound in-house. They did some experiments as well. Some of their data is shown on the right-hand side. They confirmed target engagement through SPR, so KD of 4 nanomolar. They can also confirm that this compound did not bind or have any functional activity in the structurally homologous off-target. It was very, very structurally similar off-target, and we happened to find something that was very selective. This compound was also cell active, 165 nanomolar in a cell assay that was done by the partner. And again, this was a starting point for their medchem campaign. Another example comes from a collaboration with AstraZeneca, where we ran a program with them on MERTK. We identified a number of active compounds in this project, some of which are shown in the slide here. The first 2 examples, compounds 12 and compounds 13, these come from some of our earlier libraries. Compound 12 comes from a library where it's based on SNAR and acylation chemistry. Compound 13, as you can quickly tell, comes from a dipeptide library. So first 2 cycles are amino acids and you cap with carboxylic acids. These ones were potent. If you look at the data on the left-hand side, PIC50s for these 2 were 7.6, 8.3. So they're potent, fairly selective actives. But they are amide rich. They had high polar surface area. And what our partner found out is that they had high clearance as well, which is indicated in the middle portion of this slide. Another series that they took in and looked at was compound 14, oxidizole series. This one was also quite potent. It was also very selective. And this comes from one of our more atom efficient libraries, high split size, low cycle count library. They did a number of analoguing on this compound. They found they can replace the biphenyl group on the right-hand side. They saw some improvements in solubility and potency. Sometimes they had some difficulties in sort of balancing those 2. But overall, what you can see for this hit, compound 14, it's potent, it's selective, and the clearance is quite low. So they really focused in on this series as one of their main series to focus sort of their MedChem campaign on. They did crystal structures with all of these. Compound 12 was a classic type 1 inhibitor, maybe not a surprise with the azo-indole moiety here. Compound 13 was a type 2 inhibitor. DFG was out in that crystal structure. And compound 14 was a type 1.5. So the DFG loop was in, the C helix was out. Crystal structure of compound 14 in the bind and ATP pocket shown on the bottom left-hand side, where we have this imidazopyridine ring sort of interacting in the hinge region of the kinase. The bottom portion of the slides looks at the selectivity. So partner ran a kinase selectivity panel on all of these compounds. Compound 12 was the least selective, maybe not a surprise because of the very promiscuous hinge binder motif here. But both compounds 13 and 14 were quite selective. Another project we worked on a few years ago and recently published was HAO1, hydroxy acid oxidase. This is a protein that has indications in primary hyperoxaluria. It's a rare disease. It's caused by accumulation of oxalate. So if we can inhibit HAO1, we can inhibit oxalate formation. Compound 5 on the left-hand side of the slide was an initial hit out of our DNA-Encoded Libraries. Initial biochemical activity of around 37 nanomolar. We did a few rounds of optimization on this. We wanted to improve permeability. As you can see, initial permeability was quite low, 0.82. We came out with a compound, this thiazole compound, had improved permeability as well as maintaining the potency that we were looking for HAO1. We also looked to see if we can replace the carboxylic acid. Most of the known inhibitors of HAO1 have a carboxylic acid or a very acidic group that binds in the pocket. We're hoping to try to avoid that just for further development reasons. We came up with another compound, this benzotriazole, azabenzotriazole compound. It was much weaker, but still had some good permeability. The compounds were active in a assay that we set up in-house. It's an assay that uses trogo cells and looks at the reduction of oxalate. And so what you see in this graph here is if you don't treat cells, if they don't have any glyoxylic acid treated, they're quite viable. If you add in glyoxylic acid, you can see the viability goes down. So here's just the cells with the added glyoxylic acid. And if you add in our compound number 5, you can see the dose titration. As you add in compound, we can actually restore that viability. So we see increased viability of those cells when we treat our compound in the assay. So it's inhibiting the on-target HAO1. We also got a crystal structure of this compound in HAO1. It's shown on the bottom right-hand side. The carboxylic acid of the indazole is interacting with the arginine group, classic motif for inhibitors of HAO1. And then the oxidizole with the dimethylamine moiety, shown on the left-hand side of that graph, is sort of the solvent exposed area. And this is where the library was attached to DNA. The last example I have is with KRAS G12C. So we were able to identify 3 series of covalent inhibitors through our selection process. What I've shown here are some structure blinded crystal structures of all 3 of our series bound in KRAS G12C in the GDP loaded form. So the structure that you can see, the sort of the stick figure, that's GDP bound in the GDP pocket. And then our compounds are in the green in all these crystal structures. These are bound in the switch 2 pocket. You can see they have, they bind in the same position in the switch 2 pocket, but they all have some distinct binding modes for each. 2 of these compounds, the first 2, series 1 and 2, come from peptidic libraries, including the macrocycle. But our third compound arose from a more recent atom-efficient ASA cycle library. If you compare that structure with sort of the known inhibitors, of course, really the key players in this arena, KRAS G12C, are Mirati. They have a compound in the clinic. Here's one of the examples of one of their compounds. Amgen, likewise, here example of one of their compounds. Wellspring also has a compound kind of similar in the same vein as Amgen's, but our compound was definitely structurally distinct from all of these. And if you look at the property profile of our compound versus Mirati's and Amgen's, you can see we have much lower molecular weight, much lower log P. So we're in a very good property space with this chemical series, much different than all known KRAS G12C inhibitors. We characterize these compounds quite a bit, and this slide is showing some sort of key data on these compounds. So we identified very potent time-dependent inhibition of KRAS. These compounds get around single-digit nanomolar. This is after a 2-hour incubation for a biochemical assay. They're also active in cell assays. We did phospho-ERK assay. This is sort of demonstrating the activity and downstream signaling of KRAS. We also looked at cell viability, a 5-day cell viability assay. You can see that our compound's around 30 nanomolar in those assays. Demonstrate target engagement, TM shift. So our compounds can bind KRAS, cause a thermal shift in the protein. We've confirmed target engagement with intact protein mass spec. All in all, we found that these compounds are novel, structurally distinct from anything else that's known. They're potent, very specific, and cell active for KRAS G12C. About right on time. So I guess the final remarks is the successes that we've seen really bear out in this slide here. And so what this slide is showing are really the number of licenses that we're able to provide our partners over the years. And this is what drives us. This is what we want to accomplish. We're trying to get novel and progressible equity into our partners' hands. And as you can see, as time has gone on, we've added to the licenses over time. We've been able to license compounds for a variety of target classes, receptors, kinases, enzymes. We're all listed there on the right-hand side. But we really feel this is why we're doing this, where we're looking to get novel and good equity into our partners' hands. And we hope that by making high-quality libraries, in our case is high split size, low cycle numbers, we're able to deliver on that. So I'd like to thank all of my colleagues, past and present, who have contributed to this work. And thank you for attending the conference and listening, and happy to take any questions.
Great. Well, since there are no applause, I'll say thank you very much, Chris. Fascinating talk, and really serves the importance of library design and how that influences the types of molecules you get out at the end of the day. I was curious if you could answer a quick question for our non-chemists on the call. Let's say, what's the risk of making libraries with poor chemical properties? Is there a limitation to the technology, or what would be the risk of including building blocks that put your library far out in space?
Yeah, so that's a good question, Tim. So what, I guess I have a couple answers to that. One, sometimes certain targets require beyond rule of 5 space. So, we're not saying that you shouldn't be making libraries that fall outside of that range. But some of the things you can fall into if you make sort of larger libraries is, when you are doing, if you find a hit and go into a medchem campaign, typically what happens is you're trying to find sort of the key pharmacophore for activity. So what you end up trying to do is sort of truncate that back and find the minimum thing you need to get activity and then build back out. So, by making larger libraries, you could have problems with solubility and other things down the road, but in the end, you kind of go backwards sometimes if you make things that are too large to begin with.
Okay, thank you. David, did you have a quick question before I get to the list? I see your video came on. Okay, no, great. So then, so our top upvoted question here, we have time for a couple of these, regards the application of machine learning to a Dell screen. And feel free to defer this to Jeff's talk tomorrow if it's outside your scope. Yeah. But to say that, what is the question, or what is the influence of sequencing depth on the model performance in ML? Is it a big influence?
Definitely.
No, and can you comment on where the boundaries of that might be?
Yeah, I mean, in order to support machine learning models, you got to have, quality selection data, and you got to have a lot of it. I think in the paper that I referenced, I think we make a mention of, in order to be successful in an endeavor to use machine learning, you're going to have substantial sequencing information available.
Okay.
I will
Great.
also defer to Jeff Messer.
Great. Another question is, do you have perspective on maximizing building block diversity versus incorporating some corroborating building blocks to substantiate SAR to assist in a chemical analysis? I think you covered some of that in your talk, so this may have come in a little early.
Yeah. So they're asking about incorporating building blocks to sort of aid in sort of the hit optimization aspects.
Yeah, or very similar building blocks, so that if you see something, you get some SAR out of it.
Yeah, we definitely want to include building blocks that are similar in the library. We want to see that SAR come out. One, it helps you, if you identify hit for optimization, but two, it helps you gain more confidence in the hits you're seeing out of the selection, that this is a real binding event. If you start seeing very similar building blocks showing up, it gives you more confidence that that's something, a true binding event occurring.
Great, thank you. So let's see, moving quickly through here, probably time for one more. It says, when you talk about high conversion for on DNA reactions and setting the thresholds for your building blocks, the list is moving on me. Where did that go? Where do you see can you provide some guidance on percent yields? Because even if, for instance, you take a 3-cycle library at 80% yield and use that as your dignity cutoff, putting that through 3 cycles is less than 50% yield.
Yeah, I guess, I think setting thresholds is done sort of on a library basis. I don't think we have a strict cutoff. It really depends on the chemistry. I mean, we strive to get the majority of the building blocks in that sort of 70 to 95% conversion, so that, like the viewer stated, as they're going through multiple cycles, you want to make sure that your conversions stay high throughout the time. We can't know what happens, on an individual library member's basis, but we do our best to make sure that, to be confident of the building blocks going into the library, that they will be able to do the chemistry that they can.
Okay, great. And one last one concerns the hit confirmation. And your slide about the 94% of your licensed materials coming from the design compound in the library. Wondering if you can comment on what were the other 6%?
Yeah, good question. So I guess I'll start by saying what we mean by the 94% is sort of the full, or the encoded compound. That means that the structure that the DNA encoded for is the actual compound we made and was active. So we didn't have to look at deletions or truncations in those instances. And I guess the 6% maybe, I don't know offhand, so I guess it's hard to comment, but I'm guessing it's probably, maybe instead of the full instance, it was a truncate. That was the active compound in that instance, but it's by far the minority of the cases that we encounter.
Okay, and the follow-up part of that question was, since that's 94% of licensed compounds, it's leaving out the middle part of hit confirmation from Dell. And so can you comment on how that translation from DNA sequence to confirmed hits relative to licensed compounds? And if you see an exceptionally high confirmation rate across the board, that would make it a clear indicator of library fidelity.
Yeah, that's a good question. And I'm not sure if I have a good answer for that. I'm not aware of sort of the data in that aspect, so I guess it's gonna be hard for me to answer that. Sorry about that.
Okay, no, that's fine. Just trying to get to the bottom. So there are a dozen more questions here for you that we didn't have time for. So I'll thank you for your presentation and ask you if you've got the time to address those in the chat box during the next talks.
Yes, I'll try to address those questions. Thank you, everyone.
Okay.
Thank you, Tim.
Thank you very much, Chris.